Hello again, this video is going to be about hypersensitivity. Imagine there's this guy which gets infected by bacteria or virus. The adaptive immune response provides a specific protection against infections with bacteria, viruses, parasites, or even fungi. Some immune responses, however, give rise to an excessive or inappropriate reaction. This is usually referred to as hypersensitivity. The term hypersensitivity evolved about 100 years ago, where they would sensitize an animal with antigens, like that. So the animal would produce antibodies against that antigen. Then they would expose this pre-sensitized animal with a systemic antigen. The resulting outcome was called anaphylaxis and became the term that described the immediate hypersensitivity reaction. Coombs and Gell in 1963 came up with a way to classify the different types of hypersensitivity we have. They came up with 4 different types. Type 1 was termed immediate hypersensitivity reaction and is characterized by the production of IgE antibodies against foreign proteins that are commonly present in the environment, like pollen, for example. These usually sit on mast cells. Type 2 was called antibody-mediated hypersensitivity responses, and that occurs when IgM or IgG antibodies are produced against surface antigens on the cells of the body. Type 3 is immune complex diseases, and that involves formation of immune complex in the circulation. And then the last one is type 4, and that one was called cell-mediated reactions, where T cytotoxic cell and T helper cell 1 are the main cause of that. So now let's start with type 1 and then work our way down from there. So type 1 hypersensitivity, as mentioned, is also called IgE-mediated hypersensitivity, since reactions typically rely on IgE antibody, which is nearly always going to be found on the surface of mast cells or basophils even, which in terms makes sense since the typical reactions of type 1 hypersensitivity leads to anaphylactic shock caused by an activation of these cells. This is the most common type of allergy. So there are a couple of ways to induce a type 1 hypersensitivity reaction. One of them is inhaling. Could be different types of pollen, for example, or even particles from animals, for example. Other include foods like fish or drinks like milk, or even the physical contact could induce an allergic reaction, having an IgE-mediated reaction against the things that normally don't trigger the immune response. Now, having an IgE-mediated reaction against things that normally don't trigger the immune system is usually mostly because that person has a genetic predisposition towards developing a certain allergic hypersensitivity. So let's see what happens. So let's say there's a person that has never been exposed to pollen before, right? But that person has a genetic predisposition towards developing an allergic hypersensitivity towards pollen. So let's say this person breathed in a little bit of pollen for the first time. Now let's take a look at the surface of the lungs of this person to see what happens. Beneath the surface, there are going to be cells like dendritic cells or macrophages. Now, as the person breathes in pollen, the antigen-presenting cells will find it foreign and phagocytose it and then present it on an MHC class II molecule. And then it will go to the lymph nodes through the efferent lymph vessels. And remember, the efferent lymph vessels is going to drain the interstitial fluid from different tissues in your body, right? So both the pollen and the dendritic cells are going to go through the efferent lymph vessels to the lymph node. And the lymph node looks like this, right? Or looks sort of like this. It's going to have afferent lymphatic vessels and efferent lymphatic vessels. It's also going to have medulla, it's going to have the paracortex or the T cell zone where the T cells are, and an outer cortex where the B cells are, which is composed of primary and secondary follicle. Now when they reach the lymph, what happens? Remember that the body reacts to pollen the same way as it reacts to any type of microorganisms. So it's going to produce antibodies against that pollen. So we need T helper cell 2 to make antibodies, right? So the professional antigen presenting cell will present the pollen to a naive T helper cell using MHC2, which will bind to a T cell receptor, where the CD4 will dock to the MHC2 molecule, which will cause the CD3 to induce a signal internally inside the cell. This complex of TCR MHC2 is the first activation signal to activate the naive T helper cell. Now remember, there are 3 activation signals required to activate or to differentiate the naive T helper cell. All right. The second activation signal is through CD28 and B7 molecule. And then the third signal is when it receives interleukin 4 from other T helper 2 cells or mast cells, or even basophils can also release interleukin 4. And then it will release interleukin 2, which will bind to its own surface. This is called an autocrine function. And this causes the cell to proliferate and differentiate into effector T helper cell 2, which will express CD40 ligand and will also release interleukins. Interleukin 10 suppresses the activation of T helper cell 1. Remember that T helper cell 2 and T helper cell 1 kind of works against each other. One of them proliferates, the other one is suppressed. Interleukin 4, 5, and 6 acts on B cells, and I'll show you how in a second. So while this is happening, a naive B cell that happens to have a receptor on its surface that matches the pollen undergoes a B cell receptor mediated endocytosis and expresses it on MHC type 2, right? And then it's going to travel to the secondary follicle, and then it's going to go as close to the paracortex as possible to get some help by the T helper cell 2 to proliferate and differentiate. You can find it going into the actual paracortex as well in some cases. So now, the naive B cell expresses the pollen on MHC type 2, right? It expresses it to a Th2 cell that initially got activated by an antigen-presenting cell that presented the same type of pollen. Now keep in mind that B cells are also professional antigen-presenting cells and can help differentiate naive T helper cells as well. So now remember, in the lymph node, CD40 ligand binds to CD40. This is the second activation signal for the B cell. The first being when the B cell actually binds to the pollen. So when the B cell binds to the pollen, that's the first activation signal. When the CD40 binds to CD40 ligand, that's the second activation signal. The third activation signal for the B cell to start proliferating, that is receiving interleukins from the T helper cells. And the T helper cell can release interleukin 4, it can release interleukin 5, interleukin 6, and interleukin 10, or even receive interleukin 2 or interferon gamma from Th1 cell to differentiate into a plasma cell that secretes antibodies and to memory cells. Now, keep in mind that it doesn't receive all of those interleukins together. It actually receives a specific interleukin according to what antibodies we need. So let's say we need more IgA, for instance. Then we would receive more interleukin 5. If we need IgG, we would get interferon gamma, or even interleukin 4 can get us a subtype of IgG as well. IgM is produced without the help of T cells, and IgD as well, since they are part of B cell development. But if we need IgE, remember IgE is going to be a part of the type 1 hypersensitivity. We get them by receiving interleukin 4, and they have a really high affinity to sit on the surface of mast cells and basophils. This stage of hypersensitivity is called a sensitization stage, and that stage is when you actually produce the antibodies, right? So now this guy is sensitized, right? And that means we've made antibodies against the pollen. This stage of being sensitized usually lasts from 6 months to 5 years, meaning that it is possible to lose the antibodies we actually produced for the pollen if we don't get exposed to pollen within that period, within 6 months to 5 years. And after that, after the period we're done with sensitization stage, if we have walked around for a long time without being exposed to pollen, we will lose the antibodies, and then we'll go back to not being sensitized. And then after that, once you're exposed to pollen again, you will then go through the sensitization stage again. So that's mainly how sensitization functions. So now the second stage is the second exposure. Remember, we already have mast cells with IgE type antibodies against pollen, right? Now, when this person breathes in pollen again, usually requires more than just one. It triggers the mast cells into releasing mediators like histamine, which causes vasodilation, for example, and and also increases the permeability for water and cells to go out from the blood to the interstitial space in the area where the mast cells are. This is usually good if you have an infection of a certain type of microorganisms, but in this case, when the case is only pollen, which really isn't that dangerous for the body, it's very bad because it causes unnecessary damage to your tissues. What also could happen is that it increases the permeability of blood vessels, and that causes more water to flow in, and you get more interstitial fluid than normal, and that could cause edema, for example. It also causes bronchoconstriction, so the person can't really breathe too well. Other mediators are heparin and trypsin. They have a sort of the same function. Also, it secretes out interleukin 5, which activates eosinophils and get IgA, and also tumor necrotic factor, which promotes inflammation and cause fever. So that's mostly how type 1 hypersensitivity works. It's called an immediate hypersensitivity response because once you're sensitized and you breathe in, let's say you breathe in pollen, you see all of those things that happen, they happen instantly because you already have antibodies against pollen. So that's type 1. Now over to type 2. Type 2 is what we call antibody-mediated immune response against antigens on the surface of your cells. And they're either IgM or IgG. Now remember, normally you don't really have that because you just don't want your body attacking your own cells. And there are possible reasons for this. One of them is if you're infected by complex viruses. Imagine a cell, right? And the cell gets infected by a complex virus. And complex viruses usually gets out from the cell. Once they replicate, they usually get out of the cell by what we call viral budding, right? So when they integrate their own DNA into our own genome to produce their proteins, and these proteins are usually attached on the surface of the cell, on the membrane, to prepare for viral budding when the virus actually fuses with the cell membrane to get out. And as a result from that, you tend to have antigens on the surface of the cell. What can happen is that antibodies can actually form a complex on the surface of the cell. And what do we have now? Now we have type 2 antibody-mediated hypersensitive response because you see antibodies on the surface of the cell. Other possible reasons are drug allergies. If you take any sort of drug, penicillin for example, these drugs has a tendency to also sit on the surface of the cell. If that happens and you're allergic to drugs, you also get secondary immune response. Other types are transfusion reaction. So imagine a person A having blood type A, meaning they have A antigens on the surface of their red blood cells, and another person, a person B, has a blood type B, which has B antigens on the surface of the red blood cells, but they also have anti-A, so antibodies against A antigens. So if person B is in need of blood and for some reason receives blood from person A, what then is going to happen, that anti-A is going to react to the person A's red blood cells. And now we also have type 2 hypersensitivity. The fourth way is the autoimmune reactions, for example, where we need to look at T cell tolerance. T cells get controlled so that they don't react to our own cells. And T cell tolerance happens two places. It could be central tolerance that happens in the thymus, where immature T cells are exposed to self-antigen and is killed if they do react to our own antigen. But this process isn't perfect though, and some can actually escape to the periphery, to the secondary lymphoid organs, where the peripheral tolerance happens. Here, T regulatory cells and direct inactivation happen. If it escapes from here too, we then get autoimmune disease. Now, what could happen if an antibody binds to the surface of the cell? One thing is that the classical pathway could happen, where the C1 binds to the Fc part of the antibody. Remember, these are usually IgG type antibodies. Now, C1 can cleave C4 into C4b, C4a, and C1 can cleave C2 into C2b and C2a, and these two, C4b, C2b, work as a C3 convertase, which cleaves C3 into C3A and C3B. Either it binds to the C3 convertase and becomes C5 convertase, or it binds to the surface of the cell itself and works as an opsonin. Now C5 can get cleaved. Now what can happen? You see those C2A, C4A, C3A, and C5A. These guys work as chemotaxis. And remember, these have taxi in their name, so they lead the way for white blood cells to come. This is a neutrophil, for example. They can bind to the C3b that works as an opsonin, and also, or they can bind to the antibody directly if the complement system never happened. Now, this neutrophil can either phagocyte the cell, or if the cell is too big, or if it can't phagocyte the cell, it gets frustrated and releases mediators like proteases and peroxidase to kill the cell. Another way is, C5b, C6 can bind, C7, C8, and C9. And now we get a structure called membrane attack complex. And that can make a lot of holes on the cell that ruins the internal environment of the cell. And that can cause the cell to die. Other things that could happen is, instead of a neutrophil, maybe a natural killer cell can come. Natural killer cell also have receptor for C3b and also receptors for the Fc region of an IgG. They can also release, granzymes and perforins to kill the cell. So you see type 2 antibody-mediated hypersensitivity response is more or less, it targets your own cells by a lot of reasons. So that was mainly type 2 hypersensitivity. Type 3 is what we call immune complex disease, where immune complexes like these are formed in our blood. Notice type 2 and type 3 are exactly the same. The only difference is that type 2 is related to antibodies that are bound on the surface of cells. Type 3, however, is when antibodies bind to free-floating antigens, usually in the blood. So if it's in the blood, it usually causes a systemic response, or it could be local, if you're in your lungs, for example. So let's say this is a blood vessel, right, with some immune complexes in it. Normally, your liver and spleen gets rid of these once they arrive to either one of them. But some may never even reach them. Some may get stuck on the walls of the blood vessel, right? Somewhat like this, for example. Now what can happen? The complement system can bind, just like I showed you before, and they can release chemotaxis, like neutrophils could bind and release local granules that cause tissue damage and local inflammation, and that local inflammation peaks usually in 6 to 12 hours. Even basophils can even bind, and they can release granules and vasoactive amines, and even platelets can come along and they can aggregate as a result from it. I don't know if you're familiar with the physiology of platelets, but they usually tend to aggregate on rough areas, and that's exactly what the immune complexes are once they're stuck. So that's why they aggregate on them. They could even activate mast cells in areas that can release histamine. All of this mess right here is termed Arthur's reaction by Nicholas Maurice Arthur, who discovered all of this for the type 3 hypersensitivity. The other type of reaction of type 3 hypersensitivity is what we call serum sickness disease. So imagine you're out walking and then unfortunately a snake comes and bites you and it secretes its venom into your bloodstream. And if you're quick enough and reach the closest healthcare and they luckily has an antidote, they can inject the anti-snake venom which you will have in your bloodstream. So now free immune complexes are also formed in your blood. So that is also type 3 hypersensitivity, immune complex disease. So that was type 3. Now last one is type 4 hypersensitivity, also known as cell-mediated reactions, where T helper cell 1 and T cytotoxic cell are involved mainly and can cause a lot of inflammation in the skin. One famous example is the tuberculin skin test or the Monteau's test. This test is to check if you've been infected with the Mycobacterium tuberculosis before. These bacteria release a protein called tuberculin, and if we've been infected with this bacteria before, we should react to tuberculin fast. So let's say you've never been infected with this bacteria, right? Where sensitization stage and the second exposure still counts. This tests start off by injecting a tuberculin into the skin. Langerhans cells, which is a skin dendritic cell, will phagocyte it and then run off to your afferent lymphatic vessels. So now Langerhans cells has phagocytosed the tuberculin, right? And then it's going to present it on an MHC type 2 molecule, and then it's going to travel to the lymph node to present it to a naive T helper cell. And a naive T helper cell is going to use its CD4 to kind of dock to the MHC type 2 molecule, and then CD3, which is also a part of the T cell receptor, is going to send a positive signal to the cell nucleus internally. So that is the first signal to activate and differentiate the T helper cell. The second signal is between B7 and CD28. Langerhans cells only expresses B7 once it's reached the lymph node. Keep that in mind. And then it's going to release interleukin 12. That's the third signal. Remember, look at the name. It's called cell-mediated reaction. That's why we need T helper cell 1, because T helper cell 1 help activate macrophage to induce inflammation, but they also help activate T cytotoxic cell, which is a part of the cell-mediated reactions. Alright, so we need T helper cell 1. So now after interleukin 12 has bound to the surface of the naive T helper cell, it's going to release interleukin 2, which is going to bind on its own surface. That's called an autocrine function. And that's going to help it grow and differentiate and undergo clonal expansion. So now the Th1 cell, it's an inflammatory T cell that activates macrophages by releasing interferon gamma, which also causes production of IgG and also decreases the Th2 production to focus more on the Th1. It's also going to release interleukin 2, which helps B and T cells to grow. So a dendritic cell is very good at cross-presentation. Keep that in mind, because dendritic cells are very big and they have these processes which can reach to a lot of cells and activate them simultaneously. So that's what it usually does. It's also going to present a part of the tuberculin on MHC class 1, which is going to attach to the T cytotoxic TCR receptor. Because remember, they have CD8, they don't have CD4, and CD8 helps dock to the MHC class 1 molecule. So and that's also going to induce a signal to the CD3. That's the first activation signal. The second activation signal is B7 and CD28, and then the effector T helper cell 1 is going to release interleukin 2 and interferon gamma. They both are going to help the T cytotoxic cell become an effector T cytotoxic cell, which is going to express what is called Fas ligand. And any type of cells that are infected is going to express FAS. And that's exactly why the T cytotoxic cell has FAS ligand, because it's going to bind to FAS, the receptor, and release perforins, which perforates the membrane of the cell, and granzymes, which usually fragments the DNA of the cell and causes it to die. Now remember that Th1 and T cytotoxic cells also becomes memory cells. And memory cells are the ones that can give you a reaction really fast, right? So now we've done with the sensitization stage, and if we get exposed to tuberculin again after some time, about 28 days for example, so it gets in, right? So let's say Langerhans cells gets it. It's most probably Langerhans cells that's going to meet it the first time, because in the skin you're gonna have a lot of Langerhans cells, which is the skin dendritic cells. But this time though, it doesn't really need to go to the lymph node, because a naive T helper cell and naive T cytotoxic cells have already been presented, and now we have memory cells of tuberculin. And these memory cells can either be scattered around your skin, for example, or they can be in the lymph nodes. Now, all these dendritic cells need to do is to present tuberculin to the on an MHC to the memory T cells. Th1 gets activated and releases interferon gamma that activates macrophages, which is specialized in inflammation. So this one is going to release interleukin 1, interleukin 6, and tumor necrotic factor alpha, which acts as pro-inflammatory, meaning they're going to increase the permeability of the blood vessel that causes water and also white blood cells to flow in and cause edema, which is a swelling due to too much fluid in the interstitial space. Other thing that could happen is that it could get inside the cell or even between the cells, and that could cause the effector T cytotoxic cell to actually kill the cell that are infected with the tuberculin. So I hope this video at least helped you a little bit to understand the hypersensitivity, and I hope this was helpful.